But how was the
Maybe you
session?
can help us too.
Yeah, I
Yeah.
like that he was so open to us sending questions. He was like, "Oh, please send me an email if you have any problem." This was really nice from his part.
Yeah.
Was a really nice atmosphere too. Really light. I'm not sure if that's the right word.
Yeah. So what is your remaining question, just out of interest?
Let's see. For example, we plan to do a hydroculture of tobacco. But we heard that there are some obstacles like tobacco not liking hydrocultures, pretty much.
Yeah.
That's not good.
But there are
And
protocols, right? So I think I saw some protocols for this in there somewhere, aren't there?
there's someone at our university who did it.
Mhm.
I think we contacted him or her. I think her already.
Mhm.
But there was no answer yet. I
Okay.
think.
I also
Yeah.
have no plan on like the protocol of hydroculture of tobacco, but I can imagine that it has problems with that.
Yeah. I know a
Um,
little bit about growing greens in your basement in hydroculture
Yeah.
and
because I read a lot of it, but I mean it's not optimal if the plant doesn't like
yeah,
its roots
it,
to be underwater.
yeah, like many people, many plants don't care, but like tobacco, especially, it's quite known that it does care.
Yeah.
Yeah. What's the other question?
Let's see.
We have like another problem is because we use toxic chemicals, it has to happen under the what means Abzug in English?
Fume hood.
The what?
The fume hood.
We have to work on the fume hood, and yeah, we have to build like a construct to fit our 50 plants into the fume and to bring it to like use artificial light and stuff. It will be interesting. So we have our problems, but we have ideas to solve them.
But this is not that's definitely a solvable
Yeah,
problem,
it
right?
is.
So also like if you need some like extra plant lights, these companies are very willing to like at least lend you these special grow lights. I wouldn't experiment with like shitty whatever LEDs because it's way too less light for most plants, and like you would just have like cripple tobacco growing in there if you just buy these 20-euro LED lights. But rather just write these companies if you could like get one of these really grow lights or like plant lights. Also these grow shops, like these real grow shops, you know what I'm talking about. They also like were willing in, for example, in 2019 to like give us 2 or 3 different of these grow lights. So don't hesitate to ask for
Yeah,
that.
they
What?
even offer some which can't be detected by a helicopter.
Okay, so you got quite some experience there, but like I would highly recommend to get quite some light in the fume hood then, and then I don't see a problem if you have a group which is offering you 2 or 3 fume hoods to place all these tobacco plants. Um, it shouldn't be a problem, or is it? Is it a problem?
I think we have one film.
Yeah. But you can also like put quite some tobacco plants in a very short spot, and they will still grow quite fine because I don't know.
Yeah, they're still
They are,
pretty young, so they're not that big, I guess.
yeah.
Yeah.
But like, how would you like? I understand what your project is, and I understand what the purpose of this is. But like, what again was your output of your plants detecting it? Would they like produce this fluorescent protein or like chromophore? What is the output?
They would produce Ruby, so a signal output. But our experiment with the hydroculture is just to test if the chemicals get absorbed by the plant. Our signal cascade we will test in bacteria and maybe protoplasts, also tobacco. Yeah, we just want to test if it's in there, and then can analyze some different fibers of the plant.
Yeah, and then you would do like get the tissue and do some HPLC, or what's the plan?
Yeah, that was one idea within a comparison with just a chemical, and then we should be able to compare it if it's there or not.
Yeah.
Yeah, we hope that this works.
Okay, sounds good. And then basically this is like just to show the feasibility in the real world, but then the whole detection would work in the protoplast, which is way easier to work with, right?
Yes.
Yeah, makes sense. Yeah, I think then it's even more should be even more feasible, because basically yeah, you don't have to like get these plants out and measure, for example, or like get the leaf tissue to measure the gene expression. That would be maybe more challenging then.
But it could be quite cool. I mean, you can do also transient expression in leaves, right? In tobacco leaves. So I mean, wouldn't it be possible to like transiently put your construct into some of the leaves, let them grow in this hydroculture, and see if you get an output in the leaves?
I mean, this was also a plan to put it in transiently in tobacco leaves, but I'm not sure if that was also planned for hydroculture or not.
Okay.
Not really, I think. The whole purpose of the hydroculture is to test if it gets absorbed by the roots.
Yeah,
and,
yeah, sure.
Gets in
the leaves, like transport.
Leaves, yeah. Leaves. That's the word I was seeking.
Yeah.
Just as a side note, so I never like measured light intensity inside a fume hood, but the ones in the lab I'm in, they feel quite bright, like very bright. I don't know if this might be an idea to just maybe measure like how much light is there. Maybe you don't even need so much additional lighting. If you have fume hoods where you can turn on the light.
I don't think our ones have lighting built in.
Okay.
I hope it doesn't get warm on the fume hoods, but because of the circulation, I'm quite positive.
How would you
tobacco can tolerate quite some temperature. We have some in greenhouses, and like in the, you know how hot it was in Germany in the last weeks. We had above 50 degrees Celsius in the greenhouses, and they look fine.
You wouldn't.
Yeah, maybe not for the longest time, but
yeah,
I just need to survive like 3 weeks maybe, or less.
Easy.
Is it actually in the end problematic for you to like discard the plant material because they absorb these chemicals you're working with, or like how does it work in the end?
Yeah, that's a point we thought about, and yeah, we're still thinking about it. So we have like we have like the problem that we have toxic chemicals and GMOs. So yeah, so we thought about, okay, cool. It has a very low pressure. You high. What's the name of the how
vapor pressure?
Thank you. It has a very low vapor pressure, so we could put it in the autoclave. We think so. That's the whole idea. So to kill everything, and
but
then
we also cannot be sure if it's staying in there. So we're not sure if autoclaving it is the way to go. You have to
But
activate them on another
it could be
in another way.
because if it's not dangerous, then it's okay. It's about the concentration. I think we won't use that much of
Yes.
the chemical.
Yeah,
I'm
you
not
would
sure
just
if I want to test it.
you would just
It-
have to kind of kill the plant and then discard
Yeah.
it in the chemical yeah, in the right place for the chemical waste, right?
Mhm. Yeah. But Leonard, remember, we won't open the autoclave. Lars will do it. So no. That's of course we will think about it. We have to. If
Yeah.
it doesn't work, we have to think about other solutions like using a filter. This could work, but then we have to like press it. I don't know. It's difficult. We still have to think about it.
You can put your whole plants into a blender and make plant juice and filter that.
That's our
Yeah,
stuff.
or Thermomix. It's
Yeah, I just
Are these
realized
chemical?
I just realized now that we are all from Marburg and Bielefeld, and we already kind of meet every week.
Oh.
So
yeah.
Isabella, I think you're the only one from another team. Do you have any question?
Yeah.
Or...
Not actually, no. I think I'm fine. Mine are kind of specific because I'm using tomato, and we're gonna measure things on pollen. So it's kind of hard.
From the session before, when Mark mentioned this LCMS thingy that you could try, I did a quick Google search. So there are papers where they try to analyze certain chemical compounds in pollen. Maybe you can check that out. But they used like bee pollen that you can apparently buy somewhere. I don't know how people collect that. Yeah, so I don't know how much it is. Maybe you can just check that out. But there is literature on people.
Yeah, there's a specific quantity that you have to collect to be able to measure. That's why I asked. But yeah, I'll check it out. And also here we have a huge problem that I'm from Brazil and we don't have much access to laboratory because of the pandemic. So we're kind of suffering.
Yeah, I can imagine. So I was not in the session. What's your project about, Isabella?
So we are trying to make a genetic modify plant that can be able to degrade pesticides, specifically on the pollen, so that we can preserve the life of bees while maintaining the effects of the pesticide in the remains of the plant.
Yeah. So basically, you would like to express like pesticide degrading stuff in the pollen, or?
Yeah, basically.
Yeah,
we're using a promoter specific to pollen, so the enzyme will be expressed only there.
Yeah. And you said you would work with tomatoes, or? Oh.
Why tomato?
Yeah, that's also my question.
Yeah, so that's, we were going to work with Rhodopsis, right? That's the name. So, but there's a problem that in the place we are located, it doesn't grow well, and it's kind of a problem. The lab that we are going to use for it just doesn't have, it just wouldn't work. So we took tomato because one of our PIs is working with that.
Yeah.
And it can help us with that. And it also grows fast, is economically used in Brazil a lot. So it has its benefits. And also
and
tomato,
you can eat tomatoes in the end, huh?
Yeah. And also tomatoes only have pollen and not nectar. So it's one thing we have to be less concerned about. We don't have to be measuring both of them when we are concerned about bees.
Yeah.
If I'm not mistaken, tomato also has a big problem with pesticides, right? Because it's so they use a lot of pesticides in tomato, if I'm not mistaken, because it has so many pests and stuff.
Yes, they do.
Yeah.
We talked a lot with like people that worked in the field and with crops, and they are also suggesting that we study like coffee and wheat and things like that. But we are like, we have to focus on something first. So
Yeah.
let us just try it out.
But you already talked to experts or like to like other people and for your integrated human practice, or what was your discussion about?
Yeah, we talked both for the human practice and both for so we can have some feedback on our project as well.
Yeah. Cool.
Because it's a huge thing here in Brazil. Like agriculture is a huge part of our economics. So we wanted to know exactly how it would be accepted here, like putting a GMO in the fields and everything like that. So how would the specialists recommend us doing that? How it would affect bees? Because this is also a thing that we have to consider, like what the types of bees that we are going to focus on, what kind of pesticide we are going to focus on, how it's going to degrade it, and all things like that.
Cool. I think we will kick up.
Yeah.
Oh.
We're being kicked out of the room.
Oh.
But
yeah. I clicked away. I clicked the window away. I'm not kicked out.
Recording in progress.
I think